Uh, my way of thinking about this, I'm not a regular at the group. Unfortunately, I have often a conflict at this time, but my understanding is it's somewhat informal. So my thinking about it is I'll sort of walk you through, what we're trying to accomplish. What's the basic architecture where we see ourselves kind of differentiating a little bit from other options out there, and then leave a lot of time for questions and discussion. So, just a quick intro to the team. So I think, one of the interesting aspects about our group is that we're very engineering heavy. So we have a lot of neuroscience within the group, which is basic, clinical, and computational neuroscience. But we also have mathematicians, computer scientists who are working on high performance computing, and people like Chris Rakakis, who specialize in optimizing ODE systems. His degree is actually in pharmacology, but a lot of the work that he's done actually has been in industry, developing control systems engineering type platforms for Boeing, Mitsubishi. He has actually a pharma platform known as Pumis AI. But what's interesting is that a lot of the advantages that you see in some of these platforms developed in other industries can be brought to bear within the biotech industry. And that was one of our real motivations from the start. I also want to mention that although, the co-founders originally set up the team is really run very much at this point by some fairly senior individuals who've been, doing a lot of the hard work of laying the groundwork here. Johan is at the top, but he's actually the most recent person who was hired maybe last week, thanks to Jason. And we're very pleased to have him. But he really occupies a critical role because our business model, as I'll get to the end, is forward deployed engineering. And we really need to be able to bridge, the computational modeling that people like Anand are developing with the UI UX that people like Johan Antoine Edouard are developing. And it all sort of comes together with the high performance computing. So since I think most of you are coming from the farm industry, right, or kind of adjacent to it, yes or no? It's Zoom, so it's hard to gauge yeses and nos or nods or whatever. But that's kind of my understanding. One of the real kind of differentiators for us getting going is that from the beginning, when I formed the team, I really wanted to focus on biotech. I really wanted to solve problems that were going to lead to innovation and industry. So from that perspective, there was a lot of fantastic work that's been done on platforms of many types, at the neuroimaging scale, at the, spiking neuron scale and everything in between. But unlike many of those platforms, my aim was not primarily to develop a tool for computational neuroscientists. I felt like those tools, to a very large extent, already existed. But what I really wanted to do was to develop a tool for experimentalists and the biotech industry, people who are actually trying to bridge the scales in ways that were meaningful, in terms of outcomes. And that is actually already kind of a different way of thinking about things, and it imposes certain constraints. So if we think about the pipeline for pharma, starting from small molecule to hitting, targets, which might be receptor targets or ion channel targets, to, microassemblies and circuits, all the way to biomarkers, and then ultimately to behavior. One of the difficulties with many of these platforms is they were optimized for a particular scale. So for example, in the neuroimaging world, there are a lot of what's known as computational platforms, but the focus of these is really on developing classifiers for neuroimaging data. And these classifiers might do a great job of identifying particular patterns in neuroimaging that are associated with particular clinical outcomes, but they can't say much about mechanisms. And likewise, there are a lot of tools that are really focused at the spiking neuron and small, microassembly, small circuit scale, or at the neural mass scale, which is a different scale. But what we could not find was actually something that was able to bridge the scales from small molecules all the way to outcomes. And this is actually exactly what is needed in order to make sense of what happens here, the so-called killing ground. This is not my term, but within the pharma industry, as you probably know, there's a severe drop-off in terms of where the failure rate occurs of promising compounds. And it's really the distinction between what happens at phase 1 and what happens at phase 2. And if you think about what exactly is happening during that precipitous drop from phase 1 to phase 2, it's really when those promising compounds kind of hit the wall in terms of engaging in terms of efficacy. And by efficacy, I mean clinical efficacy, not necessarily the way efficacy is sometimes used within the preclinical domain. Efficacy could be viewed mechanistically as efficacy means I'm hitting a particular target reliably, which could mean that, my binding is working or I'm crossing the blood-brain barrier. All of those could be associated with efficacy. I am triggering certain circuits to upregulate or downregulate. That could be viewed as efficacy. But in phase 2, and especially phase 3, the much harder metric is this actually remitting symptoms? Are the patients getting better? Because at the end of the day, a depressed patient doesn't really care if his receptor this versus that has been engaged. He cares if he feels better. So, in thinking about simulations, a lot of the computer-aided drug design tools that we saw that were out there were kind of engaging at this phase. But not so many tools were at this phase. And the question is why? Well, it's hard, and we'll get to that in a moment. I will say that the other approach that has been taken within the industry, because pretty much every pharma company has kind of restructured to take advantage of the, AI revolution, is the development of computational tools that are really thinking through the problem from the perspective of machine learning, primarily, and LLMs, which is let's take mechanisms on the one hand, maybe mechanisms all the way up to here, and then let's look at outcomes and let's see if we can identify relationships in an unbiased way by just taking these very large data sets that have lots and lots of information and start seeing associations. So what's the problem with that? So obviously that approach can be extremely useful. So, I think that, these approaches get, they gain traction because they do solve a problem, of course, but they also leave untouched a really important problem. And that is the problem of circularity, which is, let's say we have, a foundation model, or we're going to be looking at machine learning. And we see this all the time in terms of the neuroimaging world. The problem with machine learning and LLMs is that they have to be trained on data sets. And they can't extrapolate new information. They can see connections that might not be obvious. But at the end of the day, they're trained on measured variables. The problem is that those data sets had to make decisions, each and every one of them, as to what it was they were going to measure. And those decisions about what we measure depend on what we think is important. The problem is that we don't necessarily know what's important ahead of time. So at the time that we do a clinical trial, we have to make certain a priori decisions about what dose we're going to be measuring at, at what duration after administering the dose, and then what variables are we going to be measuring? Because we can't measure everything. And so what these methods are doing is being able to identify, again, relationships between variables, but not being able to do what-if tests, to ask counterfactual questions. What if we had done this? What if we had done that? And then the second issue is, in terms of pharmacodynamics, efficacy is the maximal pharmacological response a drug can produce at its target. But this assumes that we know what the target should be, of course, right? And so, there are many examples one could bring to bear on where these sorts of paradigms fall apart. But one of them that we're dealing with now in our group in modeling ketamine is the fact that ketamine, like so many successful CNS drugs, was a complete surprise. So, as you all know, it was really developed as a general anesthetic. And it turns out that at different doses, it goes through a regime change. It's not just that it changes from more efficacious to less efficacious. It's that it changes what the meaning of efficacious actually means. And it can be an antidepressant at one dose, a psychedelic at a different dose, a general anesthetic at the third dose, and in ways that actually require that you understand pretty complex systems, description of how different circuits are being affected and how they're interacting with each other. So if we were mining clinical data using standard clinical trials or standard QSP approaches, we likely never would have discovered ketamine's antidepressant effects, simply because nobody would have thought to have measured it at those lower doses. You wouldn't have known what they were. This was discovered by accident. And this, of course, raises the question of how many other untested drugs have completely surprising effects at non-clinical doses. And if we think about new mechanisms, do we have infrastructure in place to do all the what-if testing that could allow us to do that? Now, this problem, this what-if testing, this counterfactual testing, is the bread and butter of CAD platforms. EDA is specifically what is used in industry. Electronic design automation. This is exactly how most high stakes industries mitigate risk. They develop what's known as, in their world, digital twins. Of course, we mean something else often in the clinical neuroscience world by digital twins. But from their perspective, a digital twin is essentially a digital or computational representation of the system that allows you to poke and prod it in various ways and essentially stress test it in ways that would allow you to ask, well, what if we were to perturb it this way versus that way? And what this is not is, for example, looking at 1,000 planes coming in and landing and then doing a statistical analysis on their wing position and their landing trajectories, but instead understanding the underlying physics. So if we understand the underlying aerodynamics, then we can actually develop a digital twin that will allow us to ask about situations that we haven't observed. So that's a very powerful technique within these industries, within aerospace, for example, or the semiconductor chip industry. But, okay, so how do we do this with the thinking brain? Because usually the assumption is this is just too complex. How would we actually get to the point where we'd know the governing equations, for example, let's say of physics, but of neuroscience in this case? So that's the challenge. That's what's hard. And honestly, I mean, nobody, who's being honest can say, okay, yeah, we've solved this. But we're trying to move in that direction. And so what we're trying to do is to develop a biotech platform for in silico testing modeled on, again, these EDA platforms for aerospace, engineering, semiconductor chip industry, but thinking about it specifically for the brain and in a way that allows you to do counterfactual tests without training. And so the idea is you can run all of these mechanistic what-if tests in parallel, generate simulated outcomes across scales, and then identify the most critical preclinical experiments, optimize their design, and operationalize their outcomes. But importantly, doing so perhaps on mechanisms that haven't been tested yet and therefore haven't yet generated data. Now, all of this also needs to operate across scales, and you'll notice these are circuits, right? So this, if you look at the structure here, this is the actual GUI. This is not just a mock-up. What you can see is you've got circuits here, you've got stimuli, auditory stimuli, visual stimuli. So basically, the Neuroblox brain is designed to operate sort of like a human brain would. If we were doing a clinical trial on a human brain with neuroimaging, we might put someone in the scanner, we might give them a task, and we'd ask them to do something, and then their behavior would actually be an input to, our model of what's going on. In this case, the Neuroblox brain is actually generating outputs. It's behaving, it's making decisions, it's doing tasks, and so forth. Importantly, it can be given drugs. It can also be given devices. So we have a project with Massachusetts General Hospital with Mark Richardson's lab. He's a neurosurgeon, in finding ways to optimize deep brain stimulation with different parameters of stimulation, placing an electrode in different places. This is the kind of what-if testing that in a neurosurgical world becomes very important. And then you've got outputs on the right. So if we can change all these parameters on the left, we can simulate outputs on the right. And I want to emphasize, again, these outputs are at all scales. So we've got the raw output. Here in this case, this is ERNA, which is the output that neurosurgeons care about, showing that when they've stimulated, it's actually achieving resonance at the circuit where they need it to. And then there's the outcome that it's generating. In this case, would be tremor for Parkinson's. So I'm going to just run a quick demo here.
The Neuroblox Pharma module lets you test CNS drugs in silico. We'll show how this works using the general anesthetic, propofol. For novel compounds, we can build on this workflow to predict efficacy, identify biomarkers, and reverse engineer molecular targets. We'll first pick sites for our simulated electrophysiology. Let's record from the thalamus, inferior colliculus, and layers 2 and 3 of the anterior cortex. Neuroblox captures the spatial variation of drug targets across neuroanatomy. This allows us to more realistically predict how molecular effects propagate across circuits. For general anesthesia, the efficacy outcome we'll focus on is patients' disrupted sensory perception. To test this in silico, we'll need to present sensory stimuli. In this case, we'll use an auditory oddball task. We'll first examine baseline responses to the auditory tones. Notice the robust signal in the thalamus and anterior cortex, visible both in local field potentials and in the underlying spiking neuron dynamics that generate them. Now let's administer propofol. Again, we can observe local field potentials and their underlying neuronal activity. Importantly, they identify a biomarker for general anesthesia, one that can be measured non-invasively in human EEG. This mechanistically derived biomarker is the suppression of auditory processing in the anterior cortex, the circuit-level breakdown that underlies loss of sensory perception. Our in silico testing produced behaviors across scales, validated by real-world experiments. Crucially, our simulation arose directly from biological mechanisms rather than from fitting or training on outcome data. This means new CNS compounds or doses can be simulated even when real-world experimental data for them don't yet exist. The Neuroblox platform. Design, discover, predict.
Okay, so I think, there are some key points that I hope that this identifies. One is that we ran an experiment. We ran an experiment on the Neuroblox brain in the same way that we would do an experiment on a human brain or on a macaque brain. And we give a task. In this case, it was the oddball task. We give then a drug. We're able to see effects across all scales. And because we can do that, we can actually identify this mechanistically derived biomarker. And all of these are capabilities that would not be possible experimentally. Why? Well, because If I am doing an experiment, I have to choose where I'm taking my measurements again. So if I am doing a macaque experiment, I need to know where I'm putting my electrodes. In this case, I can choose where I put my electrodes. I can put it anywhere. I can put it everywhere. Um, and in a human, there are some places I can't visualize because I don't have the ethical ability to implant electrodes. So I have to sort of infer mechanisms without being able to see them directly. So one of the advantages, of course, in being able to establish this mechanistically derived biomarker is that we get away from the statistical problem of having, 40 of this and 40 of that and doing some statistical test on outcome data, which has a huge signal to noise problem. Instead, we can toggle the mechanism in very precise ways, see how it propagates up the scales, and then see what the impact is. What is the signature we actually should be looking for to establish target engagement? And in this case, the signature for target engagement was not the standard signature. This is not what people typically use to look at general anesthesia. But what this can do is to give you that information, which even in the case of general anesthesia is actually very useful. It allows you to get away from asking someone to count down and then hoping that that means that they can't perceive pain. In this case, you can look directly at what the brain is doing to see if it can perceive pain, and then that provides a much more objective, precise measure. Now, in terms of simulating the brain, one of the really critical design decisions is that you have to start somewhere, of course. So you can't really, design an entire brain from the bottom up, all of it at once, because it would computationally choke. There are also some aspects that are going to matter more than others in terms of our aims, which are to look at neurological and psychiatric disorders. And so we're really optimizing to begin with 3 core circuits and their interactions, which together comprise the vast majority of psychiatric and neurological disorders. And those are the corticostriatal, the corticothalamic, and the prefrontal limbic. And so, again, we had to start somewhere. What we started was actually with the corticostriatal, and developed this very sort of comprehensive validation across scales, which I'll show you in a moment, in macaques, and then extended it to healthy human adults, which I'll also show. And now we're doing a clinical trial, actually a drug trial on bipolar patients. The corticothalamic is the one that you just saw with propofol. And then the prefrontal limbic is one that we're currently under contract with a pharma company. And as you can see, does this actually cover every disorder? No, but close. If you look at down here, these are the disorders that are typically associated with each of these circuits. And of course, these are not siloed, not in the real brain and not in the Neuroblox brain either. We're developing a brain, so of course it's completely connected. I should also mention, though, something that's a little bit unique about Neuroblox is that from the beginning, we were also interested in interactions across systems. And so in this case, for example, we have the capability of looking at neurometabolic interactions, and that turns out to be really important in thinking about, for example, the age-based hypometabolism that you see in dementia. It becomes important in understanding why diabetics develop dementia earlier. It becomes important in being able to start asking what if questions about why it is that GLP-1 agonists may actually have an impact on addiction, for example. So when we do our validation, what we're doing is we're starting from the spiking neuron scale. So we look at target engagement, of course, but then this propagates up to neuronal spiking, synaptic plasticity, learning. These are not, obviously everything in between, but it's showing sort of the full spectrum of scales at which we can do validation. And again, I want to mention that importantly, because of our focus on neurotherapeutics, we make sure that we have coverage at this scale. We make sure that we have coverage at the behavioral scale, and we want to make sure that we have coverage at the neuroimaging scale. And we are also checking in between as well. So the general approach is threefold. So we build So when we are developing a circuit, we don't start from a decision that we're going to develop a circuit for propofol or a circuit for category learning. Instead, we build a brain. So if we're building a corticostriatal circuit, what we're going to do is amass and sort of vacuum in the vast amount of data sets, neuroanatomical, neurophysiological, across all scales, across the published literature, across these publicly available data sets. All of this then gets pulled in, gets integrated, and then built up according to an architecture that I'll say a little bit about. This Neuroblox brain, once it's been pulled in from the general literature and data sets, then makes a series of predictions. And the way I should also preface this is by saying that it's not one model, but multiple models, because there's some indeterminacy. When you pull in all this information, it's not that it magically converges to just one story. There are some parts that seem like they're rock solid. There's a lot of convergence between many, many groups and experiments. Then there are some parts where there's some variation, and that gives you some confidence intervals. And what you do with those confidence intervals is that you develop, parallel competing alternative models. Then what you do is you run your simulations across all scales, and then you validate against independent data. And to be truly robust, what you want to do is to validate against data that looks very different than the data that you use to develop the model. So I mentioned that in the case of the propofol circuit, for example, or the model, we did not use propofol data to develop the corticothalamic circuit. But then what we did is we gave it an oddball task. We might give it a different task. In the case of the corticostriatal circuit, we gave it a category learning task and so forth. We poke it and prod it and challenge it in various ways and see where it breaks and where it doesn't, and then validate through a systematic testing of robustness. That process is then automated. There's an extraction, an automated assembly, and a tuning that is, again, this circuit assembly is something that has started to become developed for other areas of industry in developing these EDA platforms, and we're bringing that into computational neuroscience. So what is this architecture? So again, we start with, let's say, receptor binding and its impact on the dynamics of spiking neurons. These then form inputs to microassemblies. So these microassemblies actually are of a very particular type. These are the smallest structure that complete a particular function. We call these biomimetic computational primitives. So if we are saying that the brain is processing, the question is what exactly is doing the processing? Remember that our constraint is that we want to achieve outcomes that take the form of cognition, mood, and behavior, which means that the brain needs to be able to compute. Networks will not be enough. Oscillations will not be enough. But in order to do that, there have to be computational elements within the brain. These are not necessarily microassemblies and compute functions that we discover, but these are taken from the literature or they are discovered by us. These are then the building blocks from which we create our larger scale circuits. And then those become the building blocks from which we develop our larger scale circuits. So it becomes circuits of circuits of circuits, or blocks of blocks of blocks, and that's why it's called Neuroblox. So it has a modularity that is actually optimized for a couple of things. One of them is this comparison of different model architectures, right? So I said that we're often coming up with multiple versions of the same model. And this kind of architecture that allows for modularity means that we can substitute components in a modular fashion and thus systematically look over an entire search space for many different ways that the model can be constructed. So if we look at a computational primitive, look at, again, going back to the EDA platforms that are widely used in, engineering, electrical engineering, these look at Simulink. If you wanted to set up a circuit in Simulink, what you would do is you go to your model library, you have these computational primitives. In this case, you might have capacitors or resistors or gains or filters or whatnot. And then you pull those and you compose your circuits from these BCPs or from these computational primitives. To make them BCPs, to make them biomimetic, we have to actually take the actual ones that the brain uses, as opposed to just importing electronic ones and then saying, well, they kind of approximate what the brain is doing functionally. No, we can't do this, because if we did this, actually, neurotherapeutics couldn't interact with this bottom layer correctly. Because if we put a filter, for example, that is based on an electronic filter, there's no way for a small molecule to interact with that. So here in this case, this is one such BCP. This is called lateral inhibition. You have 4 excitatory inputs. The first one that hits inhibits the rest. And here you can see that it's acting as a kind of filter, and depending on how it's tuned, that actually affects the behavior of the filter. That matters because disorders and drugs are going to potentially affect those spiking neuron dynamics. Those are inputs, of course, to these biomimetic computational primitives, which means that the compute functions operate a bit differently, which means that those effects then propagate up scales to actually affect outcomes. So here, we can look at the corticostriatal circuit. So in this case, it's doing a category learning task. So here, if you look at the stimuli, it's looking at a bunch of dots and it has to make a decision. Is it one category or a different one? Is it category A, category B? But where we have not given it what those categories are, it has to sort of figure out what they are. And you can see that when it simulates, it's actually doing computation. It's making decisions. So in the case of the propofol, which is what I showed you, if we run our simulations with the oddball task, likewise, you can see that there are computations actually happening. And this is, again, where the measured cortical activity is where we're going to look for our mechanistically derived biomarkers. This is what we can measure with neuroimaging. And I mentioned that one of the parameters we wanted to optimize over for Neuroblox is the ability to look at interaction between systems, because in the real world, comorbidities are real. I mean, it's very unusual that you wouldn't have actually systems impacting one another in lots of different ways. The metabolic module, the architect of this is Doug Rothman from Yale. I really encourage you to look up his latest paper, which is basically what he's done is he's taken a lifetime of metabolic flux equations and condensed them into the metabolic module. So this is a work in progress. He's a very wise man. It's going to take a while to download his brain, but the idea is to download all of this accumulated knowledge and to incorporate it here. Now, what does this allow you to do? So one of the things that Neuroblox can do is you can actually put it on a diet. You can put it on a ketogenic diet. You can make it diabetic. This is going to affect beta hydroxybutyrate, glucose. This affects then the TCA cycle, the behavior of mitochondria, and so forth, and ultimately neurotransmitter cycling in ways that then input, again, to our biomimetic computational primitives. So in this way, the internal variables are also functioning essentially like drugs. So, you can see how the internal milieu and also dietary external influences can affect all of these circuits as well. And again, to mention, it's not just that they're affecting the circuits, this propagates all the way up to symptoms. That's the important part. So here is an example of what this multi-scale validation looks like. So this is the output of the Neuroblox brain that was then compared at various scales to macaque electrophysiology and behavior. This is true. Again, it has to be checked at all different scales to make sure that it's not only giving you the outputs that seem reasonable, but it's getting there in ways that map onto other data. Now, one of the beautiful things about starting to integrate information across scales in a principled way, in a way that actually honors the neuroanatomy and neurophysiology, is you start to learn things that, that weren't available to you before you put all this information together. Again, because one of the dangers, of course, in doing experiments, it's not a danger. It's a natural consequences. Again, you have to make decisions about what you look at, what you don't look at. But the advantage of this in silico model is that we can look at lots of things that maybe somebody hadn't thought of looking at. And in this case, Neuroblox was able to make a neuroscience discovery. We identified a new neural code, this something called incongruent neurons, which internally we refer to as bad idea neurons. And these are neurons that fire selectively when someone is about to make an error. And there are lots of ways we might interpret what that actually means and why the brain does this. This was actually confirmed empirically. And, it was discovered in the Neuroblox brain, then confirmed empirically. But the important thing is that it actually allows for a way in principle to start doing lots of in silico experiments that otherwise wouldn't be available to people. So extending this to the human scale means that we do the same task. So here is, again, the corticostriatal task, which was category learning. We had individuals undergoing 7 Tesla fMRI and MRS. And we're looking at the same circuit, but of course, fMRI imposes certain constraints in terms of its spatial and temporal resolution. And what we found is that the circuit still operates as it does, in silico and validated in the macaque. At the human scale, the dynamics are dominated between 2 regions, the cortex and the striatum. And one of the things that I think I cannot emphasize enough is that once you start, you have the ability to look at mechanistic multi-scale computational modeling. You're surprised in ways that are that are so important, right? I mean, it's very often the case in neuroimaging that people use the term connectivity synonymously with signaling, with communication throughout the brain, right? There is an assumption that if we are looking at signaling at the LFP scale, that somehow connectivity, as measured with fMRI, is basically the same thing. It's just, passed under some particular filter. And it turns out that it's a little bit more complex than that. And in this case, it is actually exactly the opposite. And again, this gets back to the whole idea of a mechanistically derived biomarker and why it's important to know what it is you're looking for. So when we look at LFP, we see that learning increases the PFC striatal neural signaling. This is true for the Neuroblox brain, and it is also true when measured in the macaque validation. On the other hand, when we look at fMRI, learning decreases fMRI PFC striatal functional connectivity, which is a bold correlation. So again, if what you were doing is looking for an outcome, so in this case, you see a decrease in fMRI PFC striatal functional connectivity, and you say, oh, that must mean that signaling for some weird reason decreases during learning. That makes no sense. You really need to understand what is happening with the signal at every scale in order to make sense of this kind of stuff and in order to be able to make predictions that will not lead you astray. And so what is happening, just briefly, because I don't have that much time, what's happening is that when you undergo that transform between the LFP to the fMRI, you are actually changing the signal to noise in ways that are sort of really informative, actually. In that, what is happening at the fMRI scale is that there's a low frequency component that accompanies the development of those new categories. And as the brain is making a decision of, is it this category versus that category, it's actually introducing an asymmetry. And it's that asymmetry that is messing up the correlation. Because the correlations are set up in such a way that they have to be, the two regions need to be doing the same thing. And in this case, there's an asymmetry in the prefrontal cortex, but there's a symmetry in the striatal cortex. And so that disconnect is leading to what is measured as a decrease in bold correlation. Here's another kind of superpower once you have the ability to have these mechanistically derived biomarkers. Jason, you have a question?
Yeah, sorry, I was just curious, what causes the increase in the coherence?
So the increase in the coherence is an increase in signaling between the striatum and the prefrontal cortex, because what's happening is, as you're learning, the striatum is reinforcing it's a dopaminergic hit that's saying, yes, this is working or not, right? So it makes sense at the LFP scale. It doesn't make sense at the fMRI scale unless you understand kind of what's happening to the signal.
Thank you.
Okay, so here, okay, let's say we want to look at behavior. So we look at, for example, positive versus negative reward bias. Now, you would think that you could actually figure this out on a trial-by-trial basis based on behavior, right? Because you could say, all right, as the reward is positive, someone is doing one thing, and as the reward is negative, someone is doing another thing, and then we'll take all the trials that are positive and all the trials that are negative, and we'll look at a difference. You cannot do this. And the reason is that there's a fundamental confound, which is that the thing you'd use as your predictor, reward valence, is caused by the very thing you're trying to predict, how well they've learned. So in other words, when someone has learned, what that means is that they have made a decision that is correct, which means that they're going to get a positive reward. If they have not learned, then it means they're going to get a negative reward. And so there's no way to decouple those two. But, if you understand the underlying mechanism and you can simulate what a brain with positive versus negative reward bias would look like, you can propagate up all scales, identify what the signature is in the brain, and then look for that signature. And that is what we were able to do. We were able to develop a classifier based on the computational model from, again, spiking neurons all the way up to behavior. And then see that actually this was associated with a change in dopamine in the caudate, which again was predicted by the model. So the advantage of that kind of approach, of course, is that it brings you all the way to the symptom scale. Because, as it turns out, if we start thinking about psychiatric disorders as ways that are divorced from asking people, how do you feel? Like, tell me about your life. What do you think of yourself? What do you think of your situation? If you start actually observing these disorders in ways that can be understood behaviorally. So for example, in the case of anxiety disorders, this can be understood in terms of approach avoidance behavior. In the case of depression, specifically anhedonic depression, this is associated with a negative reward bias as compared to individuals both with melancholic depression as well as healthy controls. Once you have that way to understand what that signature would be for these different disorders, then of course you have a way to tailor drugs that can preferentially correct those particular disorders. Once you're able to do that, you're also able to understand the brain as a digital twin in the way that most of us hope that we could actually extend this. So not just a digital twin in the sense of an airplane, meaning the airplane behaves like the simulation says it will, but in this case, look at individual specific factors. So we're looking, for example, at genetic polymorphisms. We know that they affect the receptor gradients in ways that affect the behavior of how drugs are interacting with those receptors. That then is affecting, of course, the biomimetic computational primitives, the circuits as a whole, the circuits at large, and of course, then the behavior. So it starts to then provide a way to truly develop a pharmacogenomic screening instrument that is really individual specific and allows you to test, hundreds of medications in silico, identify what the outcomes might be, and then make a more principled decision about what therapeutic outcomes to pursue. Now, I mentioned that there's this automation component. I don't really have time to get into this. It's actually a very technical area, but very exciting one. So we normally think of AI in LLM terms, of course. That's what people are mostly using, but actually there's another area of AI known as SciML, scientific machine learning, sometimes known also as interpretable machine learning. Here's an example where you have these governing equations where you know some parts of the equation, you don't know other parts of the equation. We use then AI to then discover the parts of the equations we don't know. So it's a way to do automated circuit assembly by discovering those governing equations in a principled way. And so we've done some proof of concept. All of these methods for us were initially tested on electronic and physical systems and then extended to neural systems. And of course, this is an area of very active research by our team. So so the development of this Neuroblox brain is, when I say that there's an automation process, this is for circuit comparison, assembly, integration, and parameter estimation. And the hope is that we can move faster and faster and faster and, take advantage of these methods and really sort of extracting as much information as possible from the published literature and, again, these large-scale neuroimaging data sets. Increasingly also from pharma companies and their data sets for the questions that they're most interested in. So what is new and on the horizon as of this morning? Some of you may have noticed that Neuroblox 0.9 came out. Very exciting for us. It was really a major effort that came from a lot of beta testing. And what we learned is that we had this very cool tool. We put it in front of people and they were like, oh my God, what do I do? Where do I start? And, part of the challenge is really making this intuitive and useful for the people who are going to be using it. And what everyone said is we want a really robust, beautiful, intuitive GUI, a graphical user interface that allows us to interact with this in silico brain the way we would with a real brain, right? We want to be able to visualize it. And so this is something that is now available for the first time as of this morning. You have one-click desktop apps for Mac, Windows, and Linux, which require getting a sort of developer licenses with these various entities. As of August 1st, we will come out with Neuroblox 0.95, which is the GUI plus an IDE. So it's important to have the GUI, but it's also important to be able to interact with it. In order to be able to interact with it with a programming language, we actually invented our own programming language known as a domain-specific language. It was designed by and for neuroscientists. It's optimized for the needs of neuroscientists. It's very, very intuitive for people who work with the brain. And the GUI is kind of set up in such a way that you can code and then observe the impact of what you've just coded in the visualization in real time. So it's meant to be sort of a way to go back and forth very easily. If you want to keep up with us, I urge you to follow us on LinkedIn. You can go look at the code, neuroblox.jl. There's an installation guide there, and the documentation regarding the tutorials can also be found in this other link. So I think we are almost out of time, so I can stop here, or why don't I just stop here in case anybody has any questions.
Hi, Lily. Tony Cabal here.
Hello.
Sorry, I don't know how to raise my hand in Zoom, so I was trying to ask a question. Fantastic presentation.
Thank you.
I'll be quick. So, following your analogy of the plane, right? A 747 is a very complicated, but it's modular, right? We don't even know if we are modular, right? As entities. So how do you? So a lot of the issues that we're facing when we're dealing with failures is that we know the hallmarks of certain diseases, but we don't know the causal structure, right? We don't know how these different pieces connect with each other and which one is driving primarily the cause of the disease. So I'm just throwing that out just to get your thoughts on those issues.
Yeah, so I agree with you. The modularity is really key. And Neuroblox, as I tried to explain this blocks of blocks structure, the architecture is modular. Where it's not homogeneous. So for example, there are some modeling platforms that assume, let's say, a neural mass model, and then they just assume that neural mass model is everywhere, right? And just connected. So we don't do that because the level of specificity we're looking at in terms of circuits shows that is not the case. There's fundamental heterogeneity, but that doesn't mean that there isn't modularity. And we take advantage of that modularity in being able to set up these counterfactual alternative models, because we're able to ask, let's imagine, just to make it very simple. So let's say we had regions A, B, and C. This is a vast oversimplification, but just imagine we have a circuit. It involves A, B, and C. So what are all the ways that A, B, and C could be interacting with one another? Well, they could either be connected or not connected. If they are connected, they could be excitatory, inhibitory, or neuromodulatory. And if they are neuromodulatory, they might be neuromodulatory in different ways. And then of those, you could break this apart. And at some point, some of those combinatorial options would be not possible given what we know about physiology. So it's not that there are an infinite number of them. There are a finite number of them, and there's some indeterminacy. But then what we can do is take every one of those candidate models, run simulations. Those make predictions, and then look at the data. The data will contradict some of those models and not contradict others of those models. When the data contradict those models, we know those models are wrong. When the data do not contradict those models, we know that they're candidates. That doesn't mean that they're correct. We know that they aren't excluded. And through that process, you can then start learning. And that's the power of what this automation gives you, because if you had to do this by hand, this would be incredibly laborious. But being able to do it in a sort of an automated fashion allows you to run through thousands of options in parallel and then to start narrowing down the possibilities.
What about nonlinear feedback mechanisms, right? If you're now having these components, because that feedback, if you don't have it included in the model, you're not going to find it, right?
All of these circuits are nonlinear. All of them. So there's not a single thing in here that I would describe as linear. And the reason why, of course, is that all of these have saturation effects. All of these have feedback effects. I mean, no one is pretending this is at all linear, but you can still look at circuits. These are ODE systems. And again, if getting back to the 747, those are nonlinear too.
Yeah. But these are modular by design, right? So, which is kind of different from potentially or not, right? We don't know, but yes, I agree.
Yeah. Okay. I do have a hard stop now, but please feel free to do. No. Please reach out to me, and I'm happy to continue the conversation. Okay. Take care.
Yeah. Thank you so much, Lily.
Thank you.